Ted grew up in Orange County, and in 2015, he was accepted to Cal Poly, and he spent the next 4 years earning a Bachelor of Science in agribusiness. He approached me in spring of 2019, asking to be my graduate student, which of course I accepted, and he started the graduate program in fall of 2019. 2 years later, he's now here today to present the results of his research on hemp. So Ted, the stage is yours.
Sweet, thank you. Just give me 1 second. Can everyone see that all right?
Yes, it looks great.
Perfect, thank you, Mari. Sweet, thank you, Dr. Brown. It's been a pleasure working with you over the past couple of years, and I'm very thankful to have worked in your lab and to have the ability to research this plant. Like you mentioned, I'll be talking about my thesis project today, which is on the evaluation of masculinization treatments to produce feminized hemp seed. So let's get into it. Cannabis sativa is the scientific name of the species, but it is commonly referred to as marijuana for drug type cannabis or hemp for non-drug type cannabis. Hemp is federally classified as an agricultural commodity, and hemp is legally considered any cannabis plant with below 0.3% THC, which is tetrahydrocannabinol, the psychoactive component in cannabis. I did my project on hemp for legal reasons, but the different types of cannabis have a lot of commonalities. Hemp is naturally dioecious, meaning the female flowers and male flowers occur on separate plants. It is also a short day photoperiodic plant, meaning as the days get shorter and the nights get longer, the flowering cycle is initiated, and it is also wind pollinated. Like humans, cannabis is diploid, and the males contain heterogametic XY chromosomes, whereas females contain homogametic XX chromosomes. These are pollen sacs on a male plant known as anthers, and on the right we have female flowers with white stigmas that lead to the ovaries. In a natural population of cannabis plants, there'll be approximately 50% female and 50% male plants. But when a grower is producing cannabis for cannabinoid production, it's crucial to only have females in the field. This is because the trichomes on the female flowers contain cannabinoids, which are the compounds of interest. And having male plants in the field or greenhouse may lead to reduced yields and undesirable buds if the females are pollinated. So breeders have come up with a way to produce all female offspring known as feminized seeds. And these types of seeds are highly desirable by cannabis and hemp growers alike. The process to produce feminized seed is done by hormonally converting a female plant to produce pollen. This hormonal process is called masculinization. And as you can see on the left, a masculinized female plant produces pollen sacs in the place of female flowers. Silver thiosulfate is commonly used to masculinize hemp plants, and it is currently the standard practice. The pollen from masculinized hemp that comes from a female is homogametic with XX chromosomes, and that pollen can fertilize other female flowers that also has XX chromosomes, resulting in the production of feminized seeds. So in essence, feminized seeds are bred by crossing a masculinized female plant with a true female plant. There are some concerns with the current practices of feminized seed production because the common methods use silver. If silver, especially in the soluble form, is excessively sprayed in an outdoor field setting, it could lead to a lot of environmental toxicity concerns. So there's a need for environmentally friendly alternatives for feminized seed production, especially for field settings. This leads me to the research I conducted. There are 3 sections of my thesis. The first experiment was on masculinization, the second was on pollen storage, and the third was on the extraction of cannabinoids. Here's a slow-mo video of a masculinized hemp flower releasing pollen that then would fertilize other female flowers. The objective of the masculinization experiment was to investigate alternative treatments to masculinize female hemp plants. The process of masculinization is achieved either by introducing gibberellic acid or by inhibiting ethylene, two different plant hormones. Inhibiting ethylene is the common method to masculinize female hemp plants, and it is done by either halting the synthesis or the action of ethylene. On the right, we have the biosynthetic pathway of ethylene, where it starts with methionine, goes through a series of enzymatic processes, eventually leading to the production and synthesis, or the production and action of ethylene, where it binds to the receptor sites, causing various hormonal processes in the plant. On the left of the diagram, there are some various ethylene inhibitors, and I would like to point out a few. AVG, which is amino-ethoxyvinyl glycine, halts ethylene production by blocking ACC synthase. Another one is cobalt nitrate, which inhibits ethylene by blocking ACC oxidase. These two inhibit the production of ethylene, but there are also some that inhibit the action of ethylene by binding to the physical receptor sites. I'd like to point out 1-MCP, which is 1-methylcyclopropene, and STS, which is silver thiosulfate. Silver thiosulfate is commonly used in the hemp industry to masculinize female plants, and it is currently the standard practice. So, essentially, we compared silver thiosulfate to 3 other ethylene inhibiting treatments, as well as in combination with gibberellic acid for some of the treatments. The experiment was designed in a randomized complete block design with 8 treatments in 8 blocks. Applications occurred once a week for a total of 3 weeks, and the first application of a treatment occurred on day 30 after sowing. The variety we used for the masculinization study is called Auto Blues, which was sourced from Beacon Hemp in California. It is a auto-flowering day-neutral variety that initiates flowering irrespective of the day length. There was 2 field trials that occurred in Goleta, California, with 8 blocks and 8 treatments, totaling 64 plants per trial. And there was also a subsequent greenhouse trial in Sebastopol, California, that included 6 blocks with the most efficacious treatments based on the field trial results. This trial had a total of 24 plants. For the field trials, STS at 500 parts per million was the control. This concentration was decided by looking through the literature as well as consulting the hemp industry. There were 2 other treatments of STS at a full rate and a half rate in combination with gibberellic acid at 25 parts per million. There was also 2 treatments of cobalt nitrate, one at a full rate at 500 parts per million and another at a half rate of 250 parts per million, but that one had gibberellic acid at 25 parts per million as well. There were 2 treatments of AVG, one at a full rate of 500 parts per million and another at a half rate of 250 ppm. And there was 1 treatment of 1-MCP at 396 ppm as a spray. This concentration was recommended by the supplier, which was Smithers Oasis. We also tried applying 1-MCP as a fumigant in an airtight chamber, but didn't get any signs of masculinization. For the greenhouse trial, we also used STS at 500 parts per million as the control and used the same 2 AVG treatments as the field trial, but we added another treatment of AVG at a half rate, but sprayed twice a week for a total of 6 sprays rather than once a week for a total of 3 sprays. So we assessed the degree of masculinization by using a rating system that was adapted from Lubell and Brand back in 2018. This categorical scale uses 5 ratings of masculinization, with 1 as 0% masculinized, all female flowers, and 5 as 100% masculinized, all male flowers. It's important to note the difference between a 4 and a 5 rating. A 4 may look like a 5 with almost 100% masculinized female flowers. But if you take a closer look at this example, there's still some female flowers lingering, as you can see with the white hairs. Now on to the results of the masculinization study. In the field, 500 parts per million of STS was the most efficacious masculinization agent in this study, and the average rating was significantly different from other masculinization treatments. AVG at a full rate and a half rate were close behind, with an average rating of 3.88 for the full rate and 3.81 for the half rate. There wasn't a significant difference of treatments with AVG compared to treatments with STS in combination with gibberellic acid, but the treatments of AVG tended to masculinize the female flowers more than the treatments of STS with gibberellic acid. The treatments of cobalt nitrate and 1-MCP had ineffective masculinization and were essentially removed from further experimentation. Now looking at the greenhouse results, all treatments except for 500 parts per million resulted in the same masculinization rating. STS and AVG both at 500 parts per million averaged the same rating at 4.33, and AVG at 250 ppm sprayed twice weekly had a similar rating of 4.17. Now, here's a visual comparison of the plants treated with a full rate of STS on the left and a plant treated with a half rate of AVG on the right. As you can see, very similar masculinization with all male flowers on both of the clusters. It is important to note that the plant on the right was a standout for the AVG treated plants. Some plants look like this, but most of them had damage due to phytotoxicity. So these photos were taken a day after the first application. On the left is a phytotoxic response to 500 ppm of AVG, and in the middle is a phytotoxic response to AVG at a half rate. And on the right is a plant treated with a full rate of STS. Phytotoxicity was commonly observed in plants treated with AVG, and only a little bit of phytotoxicity was with plants treated with STS. Plants were able to bounce back relatively quickly from treatments of STS, whereas the yellowing caused by AVG lingered until flower development and pollen release. Furthermore, on the topic of phytotoxicity, as seen in figure A above, the plants treated with gibberellic acid tended to excessively elongate, causing a few plants to collapse and die, which is called lodging. In figure B, the plants treated with cobalt nitrate had some phytotoxicity and seemed to have negatively affected the plant's ability to masculinize. In figure C, AVG at a full rate caused substantial amount of phytotoxicity all the way through flower development and pollen release. These observations serve as anecdotal evidence, but more research is needed to assign how phytotoxic these treatments truly are. Regarding masculinization, STS at a full rate of 500 parts per million was the most efficacious treatment in this study and led to superior pollen production and dispersal. AVG could be an alternative to STS, but this treatment poses phytotoxicity concerns. Gibberellic acid might be effective as a synergist at a lower rate with a single application. Now we will move on to the pollen storage experiment. Here's a time-lapse video of germinating pollen tubes. To give you perspective, this video was 3 hours long, condensed to 25 seconds. And the pollen would germinate in an effort to fertilize an ovary in a receptive female flower. Now, let's get into it. The objective of this experiment was to assess the viability and storage potential of the masculinized pollen. There's multiple ways to assess pollen viability. One way is to introduce fluorescein diacetate to the grains to see enzymatic activity underneath a fluorescent microscope. This way looks to see if the cell is still doing basic physiological processes, but doesn't necessarily indicate if the grain is able to produce a pollen tube. Another way is to hand pollinate receptive female flowers to see if there's seed set. This method is hard to quantify how much of the pollen is actually viable, but could be good to see if the pollen is able to set seed after some period of storage. Another way to assess pollen viability is to germinate the grains in a germination media. We chose to germinate the pollen grains as the method to assess pollen viability because it was well established in the literature as a common practice. And personally, I think germinating pollen grains is pretty cool. There has been some recent research on the storability of cannabis pollen from a true male plant. This article looked at the short-term storage of pollen collected at various stages of pollen release. There's a 5-day period of pollen release, starting with an early stage at 62 days after sowing, with a mid stage at 64 days after sowing, mid to late at day 65, and a late at day 67. The initial germination percentages range between 30 and 50% germination and slowly decline each week after storage. The pollen collected on day 64 remained viable for 3 weeks, and it looks like the pollen could remain even more viable, or it could have been stored for longer if the study continued, as you can see with the yellow line. Another recent research article characterized the appearance and initial germination rates of pollen from masculinized female hemp plants and from true male plants. They found lower initial germination rates in masculinized pollen, ranging from 0 to 13% germination. They also looked at irregularly shaped pollen grains and found that pollen from masculinized plants had significantly more irregular pollen grains than that from true male plants. So, the methodology in the present study was to germinate the pollen to assess the viability and the pollen irregularity from plants from the masculinization experiment, then assess the storage potential of the pollen at 3 different temperatures, 22.2 degrees Celsius, 7.2 degrees Celsius, and 1.1 degrees Celsius. The protocol was to collect the pollen on day 60 after sowing. This seemed to be the day that the pollen was most available across all treatments. And then incubate the pollen in a germination medium for 18 hours at 24 degrees Celsius. Then we took pictures of the grains using an inverted microscope from Leica at 400 magnification. The ingredients for the germination medium are above, mainly consisting of sucrose at 17% and agar at 0.4%, also with calcium nitrate and boric acid. There are a lot of considerations going into assessing pollen viability, like the time of collection, the transport of pollen from the greenhouse or field to the lab, and the medium preparation before incubation. The way we scored pollen viability was by considering a grain viable if the germination tube grew twice the diameter of the grain itself. We also counted for irregularly shaped pollen grains, as you can see up here in the photo. Here are the initial germination results from the field and greenhouse trials. In the field, there were 3 treatments that produced pollen of sufficient quantities for collection. As you can see, a full rate of STS tended to produce more viable pollen grains than the initial, with an initial germination rate of 3.3% germination. There was not a significant difference between the treatments from the field trial in regards to the initial germination percentage. Plants treated with a full rate of AVG had the most irregularly shaped pollen grains from the field. So now let's look at the greenhouse trial. AVG sprayed at a half rate for 2 sprays a week tended to have pollen with the highest germination rates, but this treatment group was not significantly different from STS at a full rate and AVG at a half rate, both sprayed once a week. AVG at 500 parts per million sprayed once a week tended to have pollen with the lowest germination rates, but wasn't statistically different from STS at a full rate and AVG at a half rate. Regarding irregularity, STS at a full rate of 500 parts per million tended to have the most irregularly shaped pollen grains, but all treatment groups had a significant amount of irregularly shaped grains. So now let's look at the storage results. The average initial germination percentage of pollen from all treatments was 2.2% germination, and this pollen was from the field. After 1 week of storage in 1.1 degrees Celsius and 7.2 degrees Celsius, we saw an increase in germination percentages. For pollen stored at 1.1 degrees Celsius, there's an increase to 3.3% germination 1 week after storage. And for pollen stored at 7.2, there's an increase to 5.8% germination. The pollen from the field was able to be stored for 3 weeks or up to 3 weeks at 1.1 degrees Celsius and 7.2 degrees Celsius before showing no signs of germination at week 4. So now looking at the storage results from pollen from the greenhouse trial, the average initial germination percentage of pollen from all treatments was 3.2% germination. 1 week after storage at 1.1 degrees Celsius, there's an increase to 4.9% germination. And 1 week after storage in 7.2 degrees Celsius, there's an increase to 5.8% germination. Furthermore, there was an increase even more at 7.2 degrees Celsius at week 2 of storage, with pollen having an average viability of 7.7% germination. The pollen from the greenhouse trial stored at 1.1 degrees Celsius remained viable for 4 weeks before showing no signs of germination at week 5. So here's a visual representation of how the pollen looked each week after storage all the way up to week 5. As you can see, there is more germinability with week 0, 1, and 3, and then week 3, 0, 1, and 3, and then week 3, 4, and 5 had very little amount of germination with no germination in week 5. So wrapping up the pollen storage study, we found that pollen was able to remain viable for up to 4 weeks at 1.1 degrees Celsius. And based on the trend lines seen in both the field and greenhouse results, the pre-storage handling could be very important to accurately assess the pollen viability. Now let's get into the extraction study. Here's a picture of trichomes on a female flower. These trichomes serve as secretory vessels and are biochemical manufacturing sites for the cannabinoids. The objective of the cannabinoid study was to characterize changes in cannabinoid content after various extraction durations. The cannabinoids we looked at were CBD, which is cannabidiol, commonly associated with hemp. We also looked at CBDV, which is a minor cannabinoid gaining a lot of traction in the hemp industry. We also tested for THC, which is delta-9 tetrahydrocannabinol, like I mentioned earlier. And this compound is the notorious psychoactive cannabinoid in cannabis. Some other cannabinoids we tested for were THCV, CBG, and CBGV, as well as CBC. We looked at 2 different tissue types, leaf and flower tissues. There were 2 weight levels for the leaf samples, a 1/4 gram sample and a full gram sample, extracted for 1, 3, or 5 days. For the flower samples, we had 3 weight levels with a 1/2 gram, a full gram, and a 1.5 grams, extracted for 2, 3, 5, 7, or 10 days. And all samples were extracted in 10 milliliters of HPLC grade methanol. To test for cannabinoids, we used a Shimatsu gas chromatograph 2014 equipped with a flame ionization detector. The references for the cannabinoids were sourced from ResTech and Cayman Chemicals. There was a total of 5 injections per experimental unit, and then those 5 values were averaged to come up with a representative value for each of the samples. Here's a chromatogram of all the calibration curves for each cannabinoid. As you can see, each compound has 5 sets of peaks looking like a set of Russian dolls. These sets of calibration curves were developed from the references and were used to quantify the cannabinoids in various extraction samples. Each cannabinoid has a unique retention time, and a peak would be generated if the sample contained that cannabinoid. So a line will be drawn as a sample is being processed, and if that sample has a certain cannabinoid at a specific retention time, it would peak up and use the calibration curves to quantify it in parts per million. We were particularly interested in the ratio of CBDV to CBD, since CBDV is gaining a lot of traction in the industry. Now here's what a single calibration curve would look like. This is CBC, which is cannabichromene. Each peak in the set of peaks corresponds to a concentration in parts per million. We made a dilution series of 5 levels with 500 ppm, 250 ppm, 100 ppm, 50 ppm, and 25 ppm. This specific calibration curve had an R-square value of 0.9994. And all cannabinoids were calibrated to have R-squared values greater than 0.99. Now here's a graph of the results from the leaf extraction experiment. The total cannabinoid content for CBD and CBDV was on the y-axis. We chose to only look at these 2 compounds because they were the only 2 that were consistently present in all leaf samples. And on the x-axis, we have the extraction period in days. The 1 gram samples tested higher than the 1/4 gram samples, as expected, and there was a slight upward trend over time at the higher weight level of 1 gram. It also appears that there's more variability with more extraction time for 1 gram samples, as you can see with the box and whiskers with high variability over here. In the 1/4 gram samples, there was a slight downward trend over time, which was unexpected. Within each weight level, there wasn't a significant difference in the total cannabinoid content between day 1, 3, or 5 of extraction. In fact, it seems that full extraction was achieved only after 1 day of soaking in the methanol. Now looking at the ratios between CBDV to CBD for leaf samples, there was a common trend that the 1/4 gram samples had higher ratios of CBDV to CBD than compared to the 1 gram samples. As you can see, after 1 day of extraction, the ratio in 1/4 gram samples was more than twice than that of in the 1 gram samples. This was also seen for day 3 and 5 of extraction, with the largest gap seen on day 5, with almost a 3-to-1 ratio CBDV to CBD. Now let's look at the flower extraction results. The most variability was seen in 1.5 gram samples, followed by 1 gram samples, and the 1/2 gram samples had the least amount of variability across all extraction days. With half-gram samples, there is relatively consistent quantification over time, as you can see with the flat line from day 2 to day 10. Similar to the leaf extraction results, within each weight level, there is no difference in total cannabinoid content over time, meaning essentially you get the same extraction from day 2 to day 10. Now let's look at the ratios of CBDV to CBD in the flower tissue. There was a similar trend in the flower samples that was seen in the leaf samples, with the lower weight levels having higher ratios of CBDV to CBD. This was seen across almost all extraction days except for day 5, when there was a lot of variability with that extraction day, leading to no significant difference between the ratios, as noted with these A markers. The rest of the extraction periods had higher ratios with the lower weight level of a half gram. And it seems for both leaf and flower samples, the ratio of CBDV to CBD tends to be higher when testing with lower weight levels. Now, I want to loop back to the chromatogram I showed earlier. If we just look at the peaks for CBDV and CBD, it appears that CBDV has more sensitivity than CBD, with the higher peak for 500 parts per million than the CBD 500 parts per million. It could be that CBDV releases from the tissue more readily compared to CBD, especially at lower weights. So having lower weights when testing for cannabinoids could inadvertently lead to higher ratios of CBDV to CBD. These are speculative thoughts, but future research is needed to validate these hypotheses. So, wrapping up the extraction experiment, it is recommended to use flower samples when testing for cannabinoids because these compounds are mainly expressed during flowering. Based on these results, the flower tissues should be extracted for at least 2 days based on the present study, but a minimum extraction period would be necessary to find how quickly you can achieve full extraction. And at least 1 gram per 10 mils of methanol for flower samples would be recommended to get an accurate assessment of the ratio of CBDV to CBD. Here are some concluding thoughts from the 3 experiments. For masculinization, STS was the most efficacious treatment, but AVG could be an alternative, but poses a lot of phytotoxicity concerns. For pollen storage, pollen grains were able to be stored up to 4 weeks at 1.1 degrees Celsius. And regarding the cannabinoid extraction, the flower samples would be recommended to use, and more research is needed, especially on fresh tissue extraction. Some future research to further this work should be looking at the optimal moisture content of pollen before going into storage. Water content in pollen grains greatly influence viability, so the ideal moisture content of pollen could lead to more storage potential. Pollen germination for haploid plant culture could lead to some very interesting breeding techniques if we would be able to germinate a whole plant from the masculinized pollen. That could lead to a lot of interesting breeding techniques like double haploid fertilization. And for the cannabinoid extraction, a minimum extraction period should be investigated to see how quickly one can extract cannabinoids in flower samples for rapid throughput chromatography. And with that, I would like to thank everyone for showing up. I appreciate you listening. And I now would like to acknowledge some key individuals who helped with this research. Of course, Dr. Wyatt Brown, my advisor. He's been an incredible influence for me, a great mentor, great advisor. I'd also like to thank the rest of my graduate committee, Dr. Prince, Dr. Steinmoss, and Dr. Hoover. I'd also like to thank the HCS department and the College of Ag for such a wonderful experience the past couple of years. I am beyond grateful to be able to go to Cal Poly, and if I could do another 6 years, I would, honestly. I'd also like to thank the CSU Agricultural Research Institute for granting me a fellowship, which was very helpful, and I'm beyond blessed to have that opportunity. I'd also like to thank the CCIA, the California Crop Improvement Association, for their support. And I'd like to give a special thanks to Beacon Hemp for their incredible support throughout the past year, facilitating the research and giving me guidance throughout the whole process.